President's Day, many of our colleagues from the VA will not be able to make it today because it's a federal holiday, so some of the seats will remain empty. And maybe some of us are also observing, so we'll see. Before I introduce the speaker, let me just share with you a couple of very interesting, very exciting pieces of information. So I have started lately making you aware of recent publications from the department. And today we have a couple of very unique publications. This is from GI, looking at racial diversity and mortality and morbidity in urban patients with hepatitis C. And I see one of the authors is here, Paul. So very interesting. Thank you for this contribution. We also have another contribution from primary care or general medicine or education. There's a lot of overlap. So Diane sent me this over the weekend. And this is the how students learn through texting. So maybe one day we'll have a grand rounds about this. Another piece of interesting information is another recognition award. You probably know that oncology separated from internal medicine and is now a separate department. However, the Hem-Onc Fellowship Program is still part of our educational program. They're still part of our department. And today we recognize one of the fellows in that department, Dr. Daniel Silberger, who is actually a third-year chief fellow who has been very much involved in the restructuring the educational program at Karmanos and has been recognized by both the program director and the associate program director over there. So I'd like to recognize him. Perhaps we can give him a hand, although he's not here. Thank you. I think we did this last week. Another piece of interesting information comes from general medicine also. We have two students who are going this spring to Washington, D.C. to present this case report on osteogenesis imperfecta. These are Solhi Lee and Michael Jemail. They've been working closely with Courtney Moore, as well as Carl Lauterbach and Dr. Levine. So thank you for giving them this opportunity to shine and for this department to be recognized as a leader in academic medicine. Thank you very much. Okay, remember to claim your CME credits. So I'm not going to spend a lot of time introducing the next speaker, who is known to everybody, other than to say I'd like to express my personal gratitude and the gratitude of the department for the contributions that Dr. Madrid continues to provide to this department in all areas related to the operations of this department, whether it's clinical, whether it's leadership, whether it's scholarship and research, most importantly in mentorship. You have left marks in each and every one of these areas, and we are very grateful for your contributions. And I'm not going to take much more of your time. Thank you for being the speaker today.
Well, this is a title where we are going to talk about. So you are in the right place. It is not an oncology grand rounds. It is a rheumatology grand rounds. What would rheumatologists know about oncology anyway? So first, let's talk a little bit about the influence of chronic inflammation in promoting the formation of solid tumors. This is a very hot area in internal medicine, rheumatology, oncology. And this actually, the idea is more than 100 years old. I think Virchow developed this idea from the cell theory of that was preceding him. And he was a pathologist, and he observed that tumors often arise at the site of chronic inflammation. And he concocted the hypothesis that there was a relationship between chronic inflammation and cancer. However, nobody really paid any attention. And for 100 years, this theory was totally forgotten. But this was resurrected in the last decade or so with the finding that several types of infection, like Helicobacter pylori, is related to gastric cancer and lymphoma. Chronic inflammation in inflammatory bowel disease, of course, is related to colon cancer. The relationship between chronic prostatitis and prostate cancer is controversial, and there are pros and cons to this association. And inflammatory diseases have been related to several cancers: bladder cancer, esophageal cancer, cervical, ovarian, and thyroid cancers. So, but some cancers, some solid tumors, do not have a evident preceding inflammatory condition. Hepatitis, of course, is one of the most prominent, and this has been subjected to a lot of study. So another type of information that related inflammation and cancer has been the use of aspirin at many levels. So evidence have shown that chemopreventive effect of aspirin and other non-steroidals on colorectal cancer and probably other cancer types is important in prevention. Also, this has been used in rheumatoid arthritis for years, and this clouds the issue of incidence of cancer in rheumatoid arthritis, because it is very likely that non-steroidals and aspirin have prevented the development of some neoplasia in these patients. So there are key factors which are related in inflammation and cancer. Transcription factors like NF-kappa B, STAT3, tumor necrosis factor, and yet other recent developments, inflammatory cytokines, recently interleukin-17, and inflammatory cytokine, chemokines like CXC type chemokines, like receptor 4, and also receptor 13, which, by the way, we have cloned in breast cancer. So signaling involving inflammation, like NF-kappa B, STAT3, HIF-1 operate downstream of the oncogenic mutations that may be related to this. So activated components of the genes RAS-RAF, MYC, and RET induce tumor-promoting production of inflammatory chemokines and cytokines. So this is a relationship between the genes that may be involved in cancer and chronic inflammation. So also in the last decade or so, maybe a little bit more longer, the importance of the microenvironment has been recognized in cancer. That is, for decades and decades, the most important factor was the study of the epithelial cell, how the epithelial cell was transformed and developed into a cancer cell. But now it is increasingly recognized that cancer involves a society of cells, and the microenvironment is very important. and we'll see what type of cells we are facing in the microenvironment that may be involved in the pathogenesis of cancer. So also cancer-related inflammation can be promoted by oncogenic mutations or by other triggering events. For instance, the RAS, MYC, or RET genes, downstream, they may promote an inflammatory environment. This is called the intrinsic pathway. But inflammation or infection, and more importantly, unknown triggers are also involved in this promoting an inflammatory environment. And this is called the extrinsic pathway. This type of intrinsic versus extrinsic pathway came to the rescue of the party line, that is, indicating that mutations could be linked to chronic inflammation. But unfortunately, for the party line, this is a very small number of mutations that can do this. So by no means they explain the plethora of inflammatory conditions that are related to cancer. So this is the multi-step carcinogenesis theory, starting by the Knudsen hypothesis, the two-hit or multiple-hit hypothesis, that cancer results from the accumulation of mutations to a cell's DNA. And this leads to upsetting the normal balance between cell proliferation and cell death. Now this was a nice story that has many holes, but however was very powerful and supported research. $1 billion of research were spent looking for mutations in cancer. And this is not working. So Hannan and Weinberg produced the theory, the basis for this theory that the crucial events such as genomic instability and cell cycle deregulation, induction of telomere length, maintenance mechanism, and angiogenic switch are thought to be essential. Moreover, they outline hallmarks of cancer. These hallmarks, I think the first 6 are old, the last 2 are more recent and probably more important. So, cancer-related inflammation and adaptive immunity were really studied for a long, long time. I think from the time of Ehrlich at the beginning of the last century to Burnett later on. And this process was modified by Dunn about 10 years ago, indicating or postulating that immunoediting is responsible for both eliminating tumors and sculpting the immunogenic phenotype of tumors. So this process is thought to be mediated by cells, and no role was entertained for the autoantibody response to tumor-associated antigens. So what are these tumor-infiltrating leukocytes that form the microenvironment? And stress has been put on the left side, tumor-associated macrophages, TAMs, of the M2 phenotype. That is, the word is killing. So these are macrophages that have the ability to kill cancer cells. Also, macrophage-derived colony stimulating factor, interleukin-10, TGF-beta, VEGF, and mast cells are part of the microenvironment. However, B cells infiltrating the tumor tissue were not mentioned at all, and very little attention have been given to this. So why is the anti-cancer immune response ineffective to control cancer? Traditionally, it has been thought that the immune response in cancer is weak, is essentially weak, and is unable to eliminate cancer. However, multiple studies have shown that cancer builds a tremendous antibody response and cytokine response, which does not really control cancer. I want to mention the work of Cousin and collaborators that consistently found activated B cells in breast cancer tumor tissue. They proposed a possible role of antibody and/or cytokine-mediated effect of B cells, paradoxically promoting disease progression, that is, cancer progression. So this is part of the immune response that had been neglected for years, but now it is found that this type of response may be not favorable to control cancer, but may accelerate cancer progression. However, the studies did not indicate how this effect would be produced. So the many holes of the mutational theory of cancer led to propose alternative theories. One is the tissue organization field theory, the TOF theory of Sonnensheim and Soto several years ago, that proposed again that cancer is a society of cells and is not really related to mutational events. And here at Karmanos, in this institution, Henry Heng proposed the evolutionary mechanism of cancer, which is a very interesting theory that we may discuss a little bit further if we have time. And I would like to discuss an autoimmunity as a model for carcinogenesis, which has been suggested by our studies for the last 20 years. So autoimmunity as a pathogenic model for disease, we may consider several of these. I think there are organ-specific autoimmune diseases like Hashimoto thyroiditis, type 1 diabetes mellitus, primary biliary cirrhosis, or systemic diseases like rheumatoid arthritis, which is a prototype of the autoimmune diseases. And I put a question mark: could we consider cancer as a potential autoimmune disease? So type 1 diabetes is an organ-specific autoimmune disease that culminates in the destruction of the pancreatic beta cells, characterized histologically by islet cell inflammation and beta cell damage. These patients have a variety of autoimmune features. not only those that lead to diabetes, but also antinuclear antibodies and other autoantibodies. So this is characterized by autoantibodies to islet and or beta cell antigens, which are present at the time of diagnosis and can often be detected long before the disease. Does it sound familiar? I think this is what happens with the antibody response in the systemic autoimmune diseases like systemic lupus erythematosus and rheumatoid arthritis, in which antinuclear antibodies of several kinds, rheumatoid factors and citrullinated antibodies are present years before the development of the disease, at least of the diagnosis. So primary biliary cirrhosis is an organ-specific autoimmune disease when there is a breakdown of self-tolerance to highly conserved mitochondrial and nuclear antigens. The autoreactivity is to antigens present in the inner mitochondrial membrane. And I think it is interesting to say that we have, I'll mention later, discovered autoreactivity to mitochondrial antigens in the inner mitochondrial membrane, also in breast cancer. In primary biliary cirrhosis, the antimitochondrial antibodies recognize members of the 2-oxoacid dehydrogenase complex, which is a multi-enzyme complex. Rheumatoid arthritis is characterized by chronic inflammation that localizes in the synovial membrane, but it is a systemic disease affecting many other organs. And we stress the presence of rheumatoid factors, anti-CCP, anti-nuclear antibodies, anti-collagen antibodies, that again, they are present long time before. Now, one could ask whether is chronic inflammation sufficient to induce cancer? I think the party line is yes, probably yes, but has not been demonstrated. I don't think it is. I think it is necessary something else. I think in rheumatoid arthritis there is inflammation. Nobody could really argue against the chronic inflammatory features of rheumatoid arthritis, and yet these patients don't develop cancer except for some lymphomas and so forth. Now, the other thing that could be argued is, or mentioned against, the absolute relationship between inflammation and cancer is psoriasis. Psoriasis involves a tremendous inflammation of the skin, and yet there is no really increased incidence of cancer, skin cancer in these patients. Now, psoriasis is an H1 type disease, and it is mainly a T-cell disease and not an antibody-producing disease, not a true autoimmune disease. So the hypothesis that has governed our research for the last few years is that breast cancer and possibly other solid tumors may behave as an organ-specific autoimmune disease. So there is an interesting relationship between cancer-related inflammation and sex hormones. Levels of endogenous sex hormones are strongly associated with breast cancer risk, and this has been known since the time of Beatson, many, many decades ago. In the systemic autoimmune diseases, in practically most of them, there is a predominance of women. Women are outnumber males from 3 to 1 to 9 to 1 in lupus, for instance. And recently, we published a paper indicating that antibodies in breast cancer sera are not epiphenomena. Now, what does this mean? What this means is that autoantibodies have been described in breast cancer and all cancers known, all solid tumors, for almost a century. And no one paid any attention to them because the consensus was that they were epiphenomena, that they had no relationship with pathogenesis or carcinogenesis, and they were secondary to release of cancer antigens. So we observed a distinctive targeting antigens of mitochondria, centromeres, centrosomes, spindle apparatus, nucleoli, and stroma. So the data suggests an involvement of an entirely different set of epithelial and stromal antigens in breast cancer. So why do we think of breast cancer as an autoimmune disease? Well, both rheumatic autoimmune diseases and cancer develop after a very prolonged period of time. Antinuclear antibodies and anti-collagen antibodies are commonly found in the rheumatic diseases and in cancer. We were first to describe anti-collagen antibodies in lung cancer in 1996. Also, anti-idiotypic antibodies are found in breast cancer sera. Now, what do you think if you find this in your practice? These are anti-centromere antibodies. These are 3 different patients with breast cancer. What do you think when you find a patient with this type of pattern, which is a speckled pattern of a high titer, in your practice? You may find this in scleroderma, in some of the connective tissue diseases. So, yet you find on the arrows the centrosomes, and these are individual patients with breast cancer. So this is what we usually find in these patients. These, you can see the arrows show multiple nuclear dots and anti-mitochondrial antibodies. This is exactly what you find in primary biliary cirrhosis, except that the specificity is different. Okay, I think I will not belabor this point, but the point is that anti-centromere antibodies, anti-collagen antibodies, anti-mitochondrial antibodies, anti-histone antibodies, anti-immunoglobulin antibodies, and rheumatoid factors, we have cloned all these molecules from breast cancer tissue. Is this not what we find in the connective tissue diseases? So what is common to cancer and rheumatic diseases? So there is evidence of multiple cooperating events in the pathogenesis of rheumatic autoimmune diseases which resemble those observed in the multi-step carcinogenesis. Also, the immune response in the rheumatic diseases and breast cancer is antigen-driven. The value of antibodies for the diagnosis and prognosis of the rheumatic diseases is very well established. I think this is very common knowledge. And autoantibodies in cancer have diagnostic value. There are many publications that I haven't had time to put there, but there are many publications that establish the value of these antibodies for diagnosis. This is behaving on the contrary. So the other point is that can autoimmunity be related to tissue damage? Of course, yes. Autoimmunity is responsible for the tissue damage in the systemic autoimmune diseases, in systemic lupus, in rheumatoid arthritis, in thyroiditis. All these diseases, autoimmune attack produces tissue damage. Again, no inflammatory process is known to occur preceding breast cancer. So I want to share some experiments that we did to show this. We used the DCIS.com breast cancer model in nude mice that was developed by Fred Miller at Karmanos. That is a very useful model that repeats all phases of breast cancer in a short period of time. So the objective was to demonstrate in this mouse model that autoreactivity to human tumor antigens in this cell, which is not a cancer cell, but it is a precancerous cell, may inflict epithelial and stromal damage to the breast as the target organ, promoting chronic inflammation and cancer progression. So we wanted to demonstrate also that treatment of this mouse model with anti-inflammatory treatment and treatment curbing the B-cell reactivity could influence this process. So we used, again, the DCIS.com xenograft model, treated with dexamethasone and rituximab to inhibit the B-cell response. We did cDNA libraries with breast cancer sera, microarray analysis and sequence analysis of informative FASH. And then we studied histologically with monoclonal antibodies to cytokines, transcription factors, and we studied apoptosis. So this is the two this is on the left, we have the weight of the animals that was comparable. I think the two lower curves are the experimental, the treated animals. The difference of the curves were not significant. On the right, we have the tumor size, the tumor weight, I mean. The two upper are the controls and treated, and the two lower are the experimental. The experimental were not significantly diminished. They were diminished, but the results were not significant. So this is a type of tumor formed in these mice at 45 days. At 45 days, we have to sacrifice these animals because the tumor is very bulky and we don't want to inflict more harm in these mice. And you see the difference between the treated animals. The treated animals show slightly less reduced size tumors, but they were furrowed, irregular. I think if you see this and this, there is obviously a difference, and this were representative of the finding in many mice. This is the histology of the untreated control at 21 days, and you can see that the in situ, the DCIS, we have, this is a typical appearance with intact basement membrane and a lobulated appearance. And this is what happened in the treated animals at 21 days. You see that the characteristic DCIS appearance is totally distorted. There is maybe a little bit more vascular development here. I think this is just one slide on treating the untreated sections 21 days with TNF-alpha antibody. You can see that there is, I think, definitely involvement and on the peripheral cells adjacent to the stroma. This was also observed with NF-kappa B. In the experimental mouse at 21 days, we observed a very interesting phenomenon that was not observed in the untreated. This is a very, very large area of necrosis, and there is a thin layer of cells labeled with antibody to TNF-alpha. This is at 45 days, untreated. You can see that this looks like a solid, mature breast cancer. There is no evidence of necrosis. And this is the treated mouse, and you can see several differences. This, by the way, is in an area that had remaining tumor tissue. I'll show you more of these. And you can see that there is a vascular development, and there is a lot of connective tissue. There is a lot of connective tissue now infiltrating the tissue. Yeah, what do I do with this? How can I get to this? Eh? Tell me. Yeah, okay. At 45 days, the treatment diminished tremendously the staining for TNF-alpha. You can see remaining islets of TNF-labeled cells, but most of them have been cleared. I think we have seen this, right? Yes, it is the other way. Okay, this is the H&E on the DCS.com nude mouse xenograft at 45 days, treated with rituximab and dexamethasone. You can see the holes in this preparation. All this is necrosis, necrosis, necrosis. You see the furrows that correlate with the furrows in the macroscopic appearance of the tumor. This is what remains from the tumor. So there is a tremendous diminishing tumor mass. These are islets of remaining tumor. Islets of remaining tumor, and all this is necrotic or apoptotic tumor tissue. This is a tunnel assay showing massive intact cells with no evidence of apoptosis that would be linked to these green spots. This is in the untreated mouse. So the untreated mouse, and this is the treated mouse at 45 days. You see the tremendous amount of apoptosis developed in these tumors. This is still remaining tumor, and this is necrotic tumor or apoptotic tumor. This is another one. There is more tumor remaining here, islands of apoptosis here and there. So, in conclusion, the treatment of this mouse model with anti-inflammatory therapy and rituximab induced very marked morphologic changes in the tumor, macroscopic and microscopic. There were prominent vascular channels that were induced by treatment. And I think this is not what one would, at least with a classic theory, would look in cancer that is getting better. But there was a drastic reduction of tumor mass through marked debulking of tumor tissue. So furrowing of the tumor surface reflects marked structural changes secondary to the massive tumor loss and stromal hypertrophy. So multiple autoantibodies to centrosome, mitotic spindle particularly, and this we published this, I think Dr. Maroon published this paper. We feel that these antibodies to centrosomes and mitotic spindles are very important because they can induce chromosomal instability, which is one of the remaining hallmarks in carcinogenesis. Also, there is participation of stromal antigens in breast cancer sera. Inflammatory cytokines, TNF-alpha, NF-kappa B, interleukin-17, which I didn't show to you, are prominently involved in cancer-related inflammation and in tumor progression. Also, IgG1 and IgG3 and complement components, we showed that they are involved producing autoimmune damage in tumor tissue, sustaining smoldering inflammation. So the hypothesis is that autoimmune damage of breast epithelial and stromal cells may trigger inflammation and promote tumorigenesis. So the failure of the immune response to control breast cancer progression may be related to autoimmune damage of the breast epithelial cells and stromal cells inflicted by these changes. So chronic inflammation orchestrated by TNF-kappa B and TNF-alpha and F-kappa B could really promote carcinogenesis. So this is the type of process that we propose. I wonder if I could have a pointer. But on the left, we have a genetic damage produced by mutations, chemical, radiation, or virus. Mutations are there because they are important. They are still important. And they may be involved maybe in 10, 20% of the cases, but they are there, and they can be one of the causes. This genetic damage may lead to generation of tumor-associated antigens and stromal-associated antigens, and an autoantibody response, a B-cell response with autoantibodies, anti-centromere antibodies, anti-centrosome antibodies, anti-mitochondrial antibodies, anti-immunoglobulins, and cytokines. This society of cells would react to this autoimmune attack and will progress, and eventually an initiated cell will appear, and there will be episodes of growth and repair, and the growth and repair would be related to autoimmune attack of this society of cells. So, the experiment suggested the possibility that immunosuppressive or selective immunosuppression and anti-inflammatory treatment has the potential to contribute to the treatment of breast cancer. So, translational implications of autoimmunity damage in breast carcinogenesis, treatment of breast cancer as a chronic disease. That is, essentially, this is what we would envision to do. Inhibition of B-cell effector mechanism, inhibition of complement and anti-inflammatory therapy could potentially be helpful in reducing the need of targeted therapy or cytotoxic drugs. So effective treatment for breast cancer could potentially prolong the lifespan of patients with less adverse effects and preservation of normal lifestyle in spite of the disease. This sounds like Alice in Wonderland, and it is an expression of desire. Of course, this has to lead to further work. So there is a potential impact on the prevention, early diagnosis and clinical management of breast cancer. This is what this contribution could be. So we have published some work in the past. I think we have the first report of anti-collagen antibodies. We published more than 10 years ago in clinical cancer research, the anti-nuclear antibodies as potential markers of lung cancer. The third paper is a very important paper for me because it was suggested by a patient that I had, a patient with rheumatoid arthritis that I saw in the clinic that developed breast cancer, and I saw this patient 1 year later after she had removal of the breast cancer successfully, and for some reason which I don't remember, I repeated the ANA that was very, very high titer in the past, and it had come to almost normal levels. So since I had a register and a biobank, I went to look for the old serum, and we cloned replication protein A, the small 32 kD subunit of replication protein A from that serum. And we did ELISA, and we found that replication protein A is recognized in 10% of patients with breast cancer. In 2004, we published a paper. You can recognize some of the people here. Dr. Granda was there. Moriano was a fellow at that time and became a faculty. And Bob Carbonin was with me for several years. And we published this, a group of diagnostic biomarkers in breast cancer. Recently, we published this paper in multiple collaboration that I feel will be important. More recently, we have publications that continue to come out. So this is the list of people that have collaborated with me in the present and in the past. In the present, Marie-Claire Maroon, Judy Abrams and Wei Chen, and in the past, Lisa Pauling and Larry Tate contribute to the work that I presented, Michael Long and Frederick Miller. The people from Henry Ford, we did a project with the Henry Ford people that contributed the breast cancer sera. Carol Peebles, who worked with N. Tang, the father of autoimmunity in La Jolla. Martin Gleason from Genalite, John Tomkill, that collaborated with me at the beginning, were the people mainly involved in this work. So this is all I wanted to tell you. If you have any questions, I'll be glad to try.
I'm sure you might be also very aware that recent immunotherapy for melanoma has a significant response. And I was expecting to mention it, but you didn't mention it.
Yes, well, I think I didn't mention this because I had to prepare this in 48 hours. But this is an important thing that you mentioned. Yes. Well, thank you.